Hello there. In this video, we will be looking at one of the important microbial fermentation pathways, that is alcoholic fermentation. Now, fermentation is one among the many energy yielding mechanisms in a cell, but more than the energy yielding process, it is mainly carried out for a particular end product. In this case, we're talking about the alcohol, which is an end product of fermentation. Now, whether it is respiration or fermentation, they generally start out with an organic compound. So, we have an organic compound, for example, glucose or any other sugar which is ultimately converted to glucose, undergoes glycolysis or glycolytic pathway and then gives us pyruvic acid or pyruvate. Now, that pyruvate, its fate is determined by the presence or absence of oxygen. If the oxygen is present, then that pyruvic acid or pyruvate enters into the TCA cycle. So first pyruvate gets converted into acetyl CoA gets into the TCA cycle, then we have the electron transport chain, and that is what we call as respiration. This respiration gives us the end product such as carbon dioxide and water if it is aerobic respiration. But in the absence of oxygen, many a times, pyruvate acts as the terminal electron acceptor and undergoes a process that we call as fermentation to give us either an acid or an alcohol or a mixture of different end products. So this is the pathway that we are going to focus upon in this video. That is the fermentation pathway. Now, fermentation is a general term that is described for anaerobic because it is happening in the absence of oxygen. It is degradation of glucose because you can see here the organic compound or glucose is getting degraded or broken down, or it could be any organic nutrient to obtain energy. So in this process, we do have a certain amount of ATP being produced or energy being produced. So it is anaerobic. That's one thing to remember. It is degradation of an organic compound and it is an energy yielding process. It is usually carried out by either the facultative anaerobes or obligate anaerobes. Facultative anaerobes are those which can survive aerobically, but then if there is absence of oxygen, they do survive in the absence as well. That is, they are able to carry out the anaerobic pathway as well. They are able to survive in low oxygen concentrations as well. So examples of facultative anaerobes which show fermentation very commonly are the species of Saccharomyces, there is Streptococcus, Lactobacillus species, Clostridium species, Enterobacter, E. coli, and many, many more. So these are all organisms which carry out the fermentation pathways, different types of fermentative pathways regularly. We also have certain other yeast species, other fermenting yeast species belonging to the genera Candida or Cluveromyces, Pichia, Saccharomycodes. There are many, Torullospora, many different types of yeast species which do show alcoholic fermentation. But then we'll get into alcoholic fermentation a bit later. Now, one thing to remember is that in a fermentation pathway, there is no oxidative phosphorylation or any kind of proton gradient. This is usually seen, the oxidative phosphorylation or the proton gradient is usually seen when we have the flow of the electrons through a series of electron carriers that are embedded in the membrane. And so we call it as the electron transport chain. So at the end of the electron transport chain, there will be a terminal electron acceptor. If it is oxygen, we call that as aerobic respiration. If it is any other kind of inorganic compound, we call it as anaerobic respiration. But here, there is no oxidative phosphorylation or proton gradient. There's only glycolysis in which there is substrate level phosphorylation happening. So that is the only type of ATP production that is seen during the fermentation pathway. Now, these organisms which are carrying out fermentation, they may be facultative anaerobes or we do have certain obligate anaerobes as well. So what they do is they use this organic compound as the electron donor, that is the substrate is acting as the electron donor, and then this organic compound, usually it's pyruvate, acts as the terminal electron acceptor and gets converted either into an acid or an alcohol. One thing to remember here is that the electron donor and the electron acceptor are neutral in their redox state. That is, they are neither highly oxidized, neither highly reduced. That's why you don't have an electron flow or an electron gradient over here. So the organic substrate in fermentation is only getting partially oxidized, and therefore that is the reason only a limited amount of ATP is being produced by the substrate level phosphorylation. These are a few points to remember about fermentation pathways. Now there are different types of fates that pyruvate can end up in. That is, fermentation of pyruvate can lead to the formation of different types of end products. We can have acidic end products like lactic acid, butyric acid, or we can have propionic acid, acetic acid. There can be mixed acid formation Or we can have the production of ethyl alcohol. There can be carbon dioxide that is released. We can have the production of butanol, butanediol. So there are different end products which are being released during the fermentation pathway. But in all of these pathways, one thing to remember is that whatever NADH was being formed during glycolysis is being reoxidized during the fermentation. So in the glycolytic pathway, whatever NADH2 was being formed, that undergoes reoxidation, and then we get back our NAD, which again enters into the next round during the glycolytic pathway. So we are getting reoxidation of NADH2 during the fermentation pathways. Now, coming to alcoholic fermentation, most of the alcohol fermentation is being carried out in nature by the Saccharomyces species. Even in industry, this is the species of yeast that we use. These organisms are used to ferment sugars and convert them into ethanol and carbon dioxide. But there are certain bacterial species, some anaerobic bacterial species which also carry out alcoholic fermentation. For example, we have Zymomonas, Sarcina ventriculi, Erwinia amylovora. This is a food spoilage organism. So we do have certain other bacteria also which form or which show alcoholic fermentation along with production of acetic acid. Then they release hydrogen, sometimes they release lactate. So they have a mixture of products. And like I told you earlier, we do have other fermenting yeast species as well, which show natural alcoholic fermentation. Now, the first step of the alcoholic fermentation is glycolysis. So you need to have the conversion of glucose or the sugar into pyruvate, which is carried out by the glycolytic pathway. Now, if it is Saccharomyces, it is carried out by the EMP pathway, that is the Embden-Meyerhof-Parnas pathway, which is one of the most common pathways. But there are a few organisms which lack a key enzyme, and hence they carry out a variant of the EMP pathway called as the ED pathway. So the Entner-Diodarov pathway is carried out in this case by Zymomonas. So whatever it is, from pyruvate or from glucose, we get pyruvate at the end of glycolysis, and it is pyruvate which is now going to enter into the fermentation pathway. Now, this process of alcoholic fermentation is used in industry for baking and for brewing. In both the cases, we use different species or strains of yeast. So we have brewer's yeast, which is used for production of alcoholic beverages, and we have baker's yeast, which is used for the baking industry for raising the dough. Whatever it is usually the Saccharomyces species that is utilized. It's just that the strains are different. Now, coming to the actual process. So I told you glucose has to be converted into pyruvate. Glucose is a 6-carbon compound, like you can see over here, and then it is being converted into 2 molecules of pyruvate. So one 6-carbon compound is being converted through a series of steps in the glycolytic pathway to form a 3-carbon compound, but 2 copies of that. So we have 2 3-carbon compound molecules being formed, that is pyruvate being formed, and during this process we have production of ATP, we have production of NADH2 as well. So this is by the regular process of, like I mentioned earlier, either EMP pathway or the ED pathway. Now from here, from this step, it changes. In the absence of oxygen, pyruvate is acted upon by the enzyme pyruvate decarboxylase. Remember one thing, when it says decarboxylase, it means there is removal of carbon in the form of carbon dioxide. So you have pyruvate decarboxylase acting upon pyruvate to remove 1 molecule of carbon dioxide. Now, because we started with 2 pyruvates, hence we have 2 molecules of carbon dioxide being released. Now, when we are removing carbon dioxide from a 3-carbon molecule, obviously we get a 2-carbon molecule, that is acetaldehyde. So, pyruvate decarboxylase is decarboxylating or removing carbon dioxide from pyruvate to form acetaldehyde, which is a 2-carbon molecule. Now, acetaldehyde is then acted upon by another enzyme called as alcohol dehydrogenase, which converts the aldehyde of acetaldehyde into a alcohol group, that is ethanol. So you are just having conversion of the aldehyde group into an alcoholic group, OH group. So alcohol dehydrogenase is a big class of enzymes, and they act upon this acetaldehyde to convert it into ethanol. But it's a dehydrogenase. Now, one thing to remember about dehydrogenase enzymes, wherever you see them, is that they are involved in redox reaction. So in this case, they are oxidizing the NADH to NAD. NADH is getting oxidized to NAD, and the acetaldehyde is getting reduced to ethanol. See, if there's reduction happening over here, something else has to get oxidized, and that is the NADH. So usually when there's a dehydrogenase, it is normally coupled with a reducing equivalent like NADH, or in some cases other reducing equivalents are also utilized. So the fermentation pathway goes as such. From pyruvate we are getting acetaldehyde by a decarboxylation step, and from acetaldehyde we get ethanol, which is happening due to a redox reaction. So acetaldehyde gets reduced to form ethanol. In the process, NADH2 gets reoxidized to NAD. This NAD again goes in and helps out during the next cycle of glycolysis. So by this process, if you see, from one glucose molecule, we do get ATP during the glycolytic pathway, and we get ethanol like shown over here. Along with that, we also get carbon dioxide. So that is why we told earlier that during alcoholic fermentation, you have not only the production of ethanol, but also carbon dioxide. So these are the end products of the alcoholic fermentation. I hope this video was useful to all of you. Meet you all in the next fermentation video as well. Thank you.